Hello everyone, I'm very pleasured to introduce the article of Mycobacterial PstP impairs host RNA alternative splicing by dephosphorylation of spliceosome RBMX at S189. Mycobacterium tuberculosis is an intracellular pathogen that can survive and proliferate within host macrophages for extended periods. Tuberculosis, caused by Mtb infection, remains one of the leading infectious diseases worldwide in terms of mortality. Mtb infection significantly alters host cell signaling and protein functions, promoting immune evasion and intracellular survival. However, the molecular mechanisms underlying these interactions are not yet fully understood. In this study, we highlight three main points. First, we used a multi-omics approach, including proteomics, phosphoproteomics, transcriptomics, and interactomics, to explore the impact of Mtb infection on various host cellular processes. Second, our multi-omics analysis revealed that Mtb regulates host RNA alternative splicing by reducing the phosphorylation levels of components in the spliceosome complex. Third, we identified the Mtb protein phosphatase PstP as a key effector molecule that dephosphorylates the RBMX protein at SER189, modulating platelet-activating alternative splicing and promoting inflammation. To investigate the effects of Mtb infection, we infected THP1-derived macrophages with Mtb-H37 raw strain and performed TMT quantitative proteomics and phosphoproteomics to uncover potential dynamic changes. We observed that during Mtb infection, host phosphorylation levels were reduced, interfering with host cell signaling and immune defense processes. The phosphorylation of spliceosome proteins, which significantly regulates alternative splicing, has been widely reported. In our results, downregulated phosphoproteins were predominantly enriched in RNA splicing pathways, suggesting that Mtb might influence alternative splicing by reducing host protein phosphorylation. To further explore the Mtb infection process, we used the faster-growing Mycobacterium smegmatis to infect THP1-derived macrophages, modeling both mild and severe infection periods. Our results showed that Mtb infection downregulated the phosphorylation levels of host RNA splicing-related proteins, and likely affected spliceosome function, disrupting normal RNA splicing processes. Next, to comprehensively analyze the transcriptional changes associated with infection and determine whether changes in the phosphorylation levels of RNA splicing proteins affect host RNA alternative splicing, we performed RNA sequencing on macrophage samples infected with H37 raw. We found that Mtb infection disrupted alternative splicing of immune and cell cycle regulation-related genes. Since the expression levels of splicing-related proteins remained unchanged, these observed splicing alterations were likely due to reduced phosphorylation of spliceosome protein. Given that Mtb infection downregulated host phosphorylation levels, we hypothesized that the Mtb phosphatase PstP might be involved. We conducted proteomic and phosphoproteomic analyses to examine the targets of PstP in the host, and found that PstP is an important regulator of host protein phosphorylation during infection, particularly in splicing-related proteins. This phosphatase significantly reduced the phosphorylation levels of these proteins. To better identify substrate targets, we performed immunoprecipitation followed by mass spectrometry to identify PstP interacting proteins. We found that PstP directly interacts with RBMX and dephosphorylates the SER189 residue of this protein. This dephosphorylation significantly influences the alternativing of the platelet-activating gene. Finally, by evaluating the transcriptional levels of various inflammatory factors, we found that only the variant of platelet-activating containing exon 9 significantly enhanced the immune response, while the variant lacking exon 9 had no immune regulatory function. In conclusion, this study provides a comprehensive overview of the changes in the proteome and phosphoproteome during Mtb infection of macrophages. We observed that RNA splicing events were significantly enriched in the phosphoproteins undergoing significant changes, suggesting that splicing regulation plays a crucial role in the host response to infection. Transcriptomics. We identified dynamic alternative splicing events in immune-related genes. Combining protein interaction and phosphoproteomic data, we established that Mtb serine, threonine protein phosphatase PstP, directly dephosphorylates RBMX at SER189, leading to alternative splicing of platelet-activating. This change increases the production of the platelet-activating isoform containing exon 9, promoting the production of inflammatory factors.